In this video, we will be extracting DNA from a strawberry. Enjoy!
The Anglophone British Curriculum International School, located in Ho Chi Minh City, Vietnam, was established in 1994. The school provides enriched programs with a variety of subjects.
What is DNA? DNA stands for deoxyribonucleic acid. This is a molecule of base pairs structured as a twisted helix of genes. So what does this all mean? Well, there are 4 types of bases that make up DNA: adenine, thymine, guanine, and cytosine. A and T will always pair up with each other, as well as G and C. Different genes are responsible for making specific proteins for specific functions, creating the characteristics of an organism. In simple words, DNA is a long strand of different sets of instructions, all combined into a long twisted ladder inside the nucleus of a cell. These are the pieces of equipment required for the experiment.
Method. Step 1. The extraction solution needs to be made by mixing 90 ml of water with 10 ml of soap and 1/8 of a teaspoon of salt. Step 2. Add 20 ml of the extraction solution and 1 strawberry in a mortar. Next, use a pestle to crush the strawberries well for 5 minutes. Step 3. Place the cheesecloth inside of a funnel and put this into a test tube and pour the mixture of strawberries and extraction solution through this. Step 4. Gently squeeze the cheesecloth to make sure that all the liquid passes through the cloth. Step 5. Add 5 ml of 70% isopropyl alcohol into the test tube. Step 6. Wait for the DNA strands to precipitate at the top. Step 7. Extract the precipitate using a pipette and move it to a petri dish or to another test tube.
Explanation of the method used. So, why do the strawberries need to be crushed and grinded? This physically breaks the cell walls. Why does the extraction solution need to have detergent? After mechanically breaking down the cell walls of the strawberries, the detergent is needed to destroy the cell and nuclear membranes to release the DNA. This is because it dissolves the lipids and proteins which hold the membranes together.
Without the addition of isopropyl alcohol, DNA will dissolve in water. The negatively charged phosphate groups found in DNA bonds with positively charged hydrogen atoms in the water molecules. Salt blocks this bonding because when salt is dissolved, positively charged sodium ions neutralize the negative charge of DNA's phosphate groups. To help this neutralization, isopropyl alcohol is needed.